We are going to be looking at the biological membrane. The what? Biological membrane. Now, you agree with me, as a human being you are you multicellular or unicellular? Speak for yourself. You are multicellular. Good. It means you have many cells, right? We still have unicellular organisms that have just one cell. Now, whether you are multicellular or you are unicellular, so long as you are a eukaryote, you have membrane-bound nucleus and organelles, right? Yeah. Now, let's say this is your cell. And as this is your cell, this is a nucleus. And there is a fence here surrounding the cell. Now, as a human being, as an animal you are, do you... Are you not in the animal kingdom? Are you in the plant kingdom? Now, your cell, does it have cell wall? No. It does not have cell wall, right? But we know plants have cell wall from our basic knowledge of biology, is it not? Now, what we have here is the cell membrane, is it not? Yeah. The cell membrane. Now, we want to study this cell membrane, this thing surrounding the cell. We want to study it in details. Now, the cell membrane is what is separating the inside of the cell from the extracellular matrix, from the outside of the cell, is it not? And it is not just separating the inside from the outside, but it also serves as a semi-permeable membrane, regulating what is coming in and what is going out of the cell. So you see the function is 7 here. We will look at the functions in details. But you see what is doing here, it regulates what is coming in and what is going out of the cell. And at the same time, it is protecting the inside from the outside environment. So let me take this membrane. So many persons, they've studied the cell membrane and proposed their own structure of the cell membrane. But the most accepted model of the cell membrane is the what? The Fluid Mosaic Model. There are other models of the cell membrane, but the most accepted, which is the one we're going to be studying in this course here, is the Fluid Mosaic Model. Now, I want us to analyze these two words, fluid and mosaic. When they say something is fluid state, is it static or is flowing? And it's not standing in one place, right? If something is fluid, it's not static, it's not standing. Is it not? Water is a fluid, right? Gas is a fluid, right? Solid, is it a fluid? No. Some of you do not know that gas is a fluid. Know that one today. Alright, so now this model, this Fluid Mosaic Model by Nicholson and Singer, proposed that the cell membrane is in a fluid state, it's not static. That this cell membrane you're seeing here is not like a fence standing in one place. And as it is in a fluid state, said the next thing here we see here is mosaic. When they say this is mosaic, what does it mean? Different components combined together, right? And they say mosaic is not pure, it's not one thing. We have different parts, different components. So when they say mosaic structure, different components combined together. So the fluid mosaic structure of the cell membrane, we have different parts, different molecules combined together. And as they are combined together, is it static? It's not static, right? Good. Now look at the cell membrane. According to the Fluid Mosaic Model, the cell membrane have lipid bilayer. Please pay great attention here. Because when you're writing exam, you don't write by cramming. If you understand, you see that you've written enough. Enough sense, not enough. Now, if you look at this cell membrane, it has a lipid bilayer. When they say bilayer, bi means what? Two, right? If you look at this, this is one layer. This another layer. Is it not? And I'm using something like this to represent one of the lipid. Like that. Please don't count. You say 1, 2, 3, big. 1, 2, big. No, I didn't count it. I just do. Now, this lipid now, lipid bilayer. Let me just tell you the names of the components. Then I take them one after the other to explain. So we have a lipid bilayer. Now we have a protein here and we have another protein here. This protein found here is called the integral protein. Now look at this. The integral protein, it is called integral, as you can see. Integrated, put into, emerged in. Now the integral protein transverse the two layers. The two bilayer. Are you seeing the bilayer? You can see that the integral protein is passing through the two layers, right? If you don't call it integral protein, you can also call it intrinsic protein, or you call it transmembrane protein. Transmembrane is passing through the membrane. So this transmembrane protein is actually from the outside to the inside. Do you get? We'll see the function. But the integral protein transfers the two layers. Then we have another one here, peripheral protein. The peripheral protein is just hanging on one side. It's not holding, it's not very strong. So it can actually fall out. But it's just on the surface, the cytosolic surface of one of the layers. So that is the peripheral protein. If you don't want to call it peripheral protein, you call it extrinsic protein. Intrinsic, inside. Extrinsic, on top. Outside. Just like internal and external. Alright. Then we have cholesterol. Cholesterol is doing a great job here. We said this thing is in a fluid state, right? What is responsible for that fluidity? For that fluidity of this membrane is what? Cholesterol. We'll see how cholesterol is responsible for that. But for now, just know that what is keeping this stuff here in the fluid state is what? Cholesterol. Cholesterol. Then we have glycolipid. We still have glycoproteins. We have other parts, but these are the major parts of the cell membrane. Now, I said we have a lipid bilayer here. One thing you should notice is that the lipid found here is phospholipid. The lipid found in the membrane is what? Phospholipid, right? Good. Now let's do a little revision. We have how many macromolecules? 4, right? 4. This side, give me 1. Carbohydrate. This side, give me 1. Lipids. This side, give me 1. Proteins. This side, give me 1. Nucleic acid. Thank you. So those are the 4 macromolecules. You mentioned lipids, right? Now, lipids. We have simple lipids. Please, if you want to get background knowledge of lipid, everything you need to know on lipid, I made a video on everything you need to know on lipid, and I place it in one playlist. So it's on my channel. So go to my channel, and when you get there, don't forget to subscribe. All right, now the lipid found in the fluid mosaic model is what? Is phospholipid, right? Phospholipid. Now we have what we call simple lipids. We have compound or complex lipids, and we have derived lipids. Still have miscellaneous lipids. Good. Simple lipids contains only fatty acid and an alcohol. Fatty acid and what? Alcohol. That is simple lipid. The alcohol can either be glycerol or sphingosine. All right. We are going to do this in details in 402. All right. So now, phospholipids now contains fatty acid and alcohol. Now look at this. Phospholipid. Now pay attention here. The lipid here is a simple lipid. Then we attach phosphate. Phospholipid contains what? Fatty acid, right? What else? Alcohol and what? Phosphate group. And phosphate group. So these are the 3 things found. Now, if the alcohol present in this phospholipid is glycerol, then the phospholipid is called glycerophospholipid. If the alcohol present is sphingosine, we call it what? Single phospholipid. Single phospholipid. Good. Now, so we have glycerophospholipid and single phospholipid as the phospholipids we have. But now, our point of interest here is this. It's not a mistake for us to represent this as head and two chain. Let me draw something here. Amanda. Now, this is how phospholipids look like. Don't bother about knowing how to draw them now. If you want to know how to draw them, we are going to do that later. It's already on my channel too, but we are going to still do it. Now, but in this course, in this topic, you must not know how to draw it. Now, look at this. This is the glycerol. This is one fatty acid chain. This is another fatty acid chain, and this phosphate group. So how many fatty acid chain do we have? 2, right? 1, 2, then phosphate group. Why we are representing it like this? This thing here, this head here, is the phosphate group. Then this 2 line here is the 2-T, the 2 fatty acid T. Now, is fatty acid soluble in water or is not soluble in water? Your oil, fat, your oil, is it soluble in water? It's not soluble in water, right? Since it's not soluble in water, is it hydrophobic or hydrophilic? Hydrophobic. Some of it is called a hydro. It just knows it's hydro something. So it's hydrophobic. Now, we've seen that the T, since this is fatty acid, is hydrophobic, right? But the head, phosphate group, is polar, is soluble in water. So now, like dissolves like. Fatty acid, the cucumber know that they cannot dissolve in water. So all they do, the fatty acids now, the T will now be hiding themselves inside. Because they are hiding from water in the outside and inside of the cell. So all the fatty acid T's come inside because they are hydrophobic. But the head, phosphate group, they are soluble, so they expose themselves. This is outside of the cell. This is inside of the cell. Why the hydrophobic T's hide themselves? So the head, is it hydrophobic or hydrophilic? Hydrophilic. Why the T is what? Hydrophobic. Now, that's that about the phospholipid. So the phospholipid, we say the phospholipid can either be glycerophospholipid or what? Single phospholipid. Single phospholipid. Alright, so the integral protein here, we say it transverse the 2 layers. And you agree with me that if this thing is interacting with the outside and inside, it can actually transport something from outside to inside or from inside to outside, right? These transmembrane or integral proteins, they are involved in transport. There are some molecules that cannot get into the cell on their own. They need somebody to transport them in. That is where the integral protein comes in. Peripheral protein, can they transport? They are not. They are only on the surface. They are not penetrating. So what they do is they help in signal transduction. They have mebo waiting them top. It will go tell people where they outside because it's close to the gate. You just be hearing. What did they say? Once you hear the information, it will transduce the signal to the outside. Now waiting them talk. Oh, man, I know come today. So the peripheral protein is involved in signal transduction. The integral protein is involved in transport of molecules. All right, so basically, this is this. We still have more to say about this. But for now, any question? If you're asked to draw, you don't have to cram anything. Just draw your lipid bilayer, put integral protein, peripheral protein, cholesterol. That's very important. Even if you don't put glycoprotein, no problem. But you can put. All right, so basically that's that. All right, now we've seen that the membrane is a fluid mosaic structure, meaning that the membrane is in motion. Now, what are some of the factors that can affect the fluidity of the membrane? What are some things that can make it more fluidy? And what are some things that can make it more viscous, less fluidy? Now, one factor, please you see this in an exam. One factor affecting fluidity of the membrane is cholesterol content. Cholesterol content. Now, cholesterol, if cholesterol is high, what will happen to the fluidity of the membrane? Will the fluidity be high or low? Will it be more fluidy or less fluidy? If the cholesterol is high, fluidity will be what? Decrease, right? If cholesterol is low, fluidity will be what? Increase. So you see that the relationship between cholesterol and concentration of cholesterol and fluidity, are they directly proportional, inversely proportional? Inversely, right? Meaning as cholesterol is increasing, the fluidity will be what? Reducing. While if cholesterol is reducing, the fluidity will what? Increase. But this property of cholesterol is determined or is affected by temperature. Now, increase in temperature, you agree with me that if you heat it, the molecules will acquire more kinetic energy, right? If you increase temperature, what will happen? Will the fluidity be more or less? Will the fluidity increase or reduce? Will increase. Now, take for example, you have your solid. If you heat it, what will happen? The molecules of that solid will acquire more energy. Entropy will be high, and so they'll start to move, right? Converting the solid to what? Liquid, melting. If you continue to heat, the molecules will still acquire more kinetic energy, and they'll start to move far apart into the gaseous state, right? So you see that heating will make it more fluid, more liquid, is it not? So increase in temperature. If temperature is increasing, fluidity will be what? Increasing, right? If temperature is decreasing, fluidity will be what? So at lower temperature, the membrane exists as a viscous substance. All right. Now that's that. Then lipid composition. What type of fatty? We know with the lipid is phospholipid. But now, the phospholipid, we have two chains. We have two tails, right? Then the phosphate head. What kind of fatty acid are we using? Is it long chain fatty acid or short chain fatty acid? Is it saturated fatty acid or unsaturated fatty acid? Now, if we use long chain fatty acids, if the composition of that lipid is long chain fatty acids with saturated bonds, will it increase fluidity or reduce fluidity? Long chain fatty acids will reduce fluidity, right? While shorter chain fatty acids will increase fluidity. Is it not? So long chain fatty acids, if you are using long chain fatty acid, the fluidity will reduce. But if the composition is short chain fatty acids, the shorter, the better. The more fluid it is. Alright. Now the types of bonds present. The more packed together the molecule is, the less fluid it will be, right? The more packed together it is, the less fluid it will be. So that's that. So basically these are the three main factors, but this is a minor one also, part of it. So don't forget that cholesterol, the more the cholesterol, the what? Okay, look at cholesterol like Gary. You are pouring Gary on something that was fluidy. The more you pour, the more solid it will become, right? The less fluidy. Is it not? The lesser the stuff or your thickener in your soup. Perfect example for people that used to enter kitchen. Your thickener, the more you put, the more it will be thick, right? The less you put, the less it will be. Right. Now, temperature, the higher you put the temperature, the more fluid. Long chain fatty acids decrease and short chain fatty acids increase. So let's look at the functions of the cell membrane. Are we together? Functions of the cell membrane. Now first, we know that the cell membrane is serving like as a barrier, right? regulating what is coming in or going out of the cell. Now the first one here is selective permeability. The cell membrane is selectively permeable. It allows non-charged molecules, substances that does not have charge, it allows them in while preventing those ones that have charge from passing. So it allows some molecules in and prevent others. That's why we say it's selectively permeable. It's permeable to some and it's not permeable to some. Allows some in while preventing others. So those things the cell needs, allows it in. But there are some other things that the cell needs that cannot pass on their own into the membrane. That is why we have ionophores, ion carriers. We'll look at that. Ion channels, we have that too. All right. But one function here is selectively permeable. Now, closure to other organelles. It serves as closure. Now, look at this. It's just like a fence housing other organelles. Just imagine you don't have cell membrane. It means you'll just be sitting and one day mitochondria will just come outside. All right. So it serves as a closure, protecting other organelles, right? So it encloses other organelles, preventing them from physical or chemical damage. That's that. Then signal transduction. You agree with me that the cell membrane, we have peripheral proteins, right? We have glycolipids also that are involved in signal transduction, transducing signals in and out of the cell. So cell to cell communication. Receptors. This cell membrane houses different receptors. So different receptors are found here. Like a receptor can, when we talk about secondary messengers, you've heard of cyclic AMP, right? When we talked about mechanism of hormone action, and I talked about fixed receptor mechanism. Now note that receptors are found in this cell membrane. Channels, just like I said earlier, there are some things that cannot pass through this membrane itself. It needs a transporter. It needs a channel to pass. Now we have channels that open for molecules to pass through. The transmembrane protein here is here also to transport molecules, right? But we have ion channels. Those channels that can transport ions. You know, it's only non-charged molecules that can actually pass through freely. But charged molecules will need a channel they can pass through. And that's where ionophores come in. We are going to take ionophores in our next topic. Alright, then cell recognition. They are involved in cell-to-cell recognition. And it is from here you can identify the cell membrane. You can identify the ABO blood grouping system. You know, the ABO blood grouping system is just based on antigen present. If the antigen present on your red blood cell. Now when we're talking about the cell, you know the different types of cell. If the antigen present on your red blood cell is antigen A, then your blood group what? A. If it is antigen B, your blood group B. If you have both antigen A and antigen B on the surface of that red blood cell, we say your blood group what? AB. But if you don't have any antigen on that red blood cell, we said you have what? Blood group what? O. So it helps in cell-to-cell recognition. So these are some of the functions of the cell membrane.